Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 16th November 2020. As we are all familiar, we are into the revision mode. We are revising chapter number 8, cell structure and functions. In our previous session, we were discussing and reflecting upon the all-important cell organelles of the cell, that is the powerhouse of the cell, mitochondria, and the kitchen of the cell, chloroplast or plastids in general. Now, in this session, we are going to talk about another important structure called centrosome. So, centrosome is an organelle containing two cylindrical structures called centrioles. And as all of you know, centrioles play very important role during cell division. If you recollect the mitosis or meiosis that we talked, there will be chromosomes in the metaphase in the center of the plate, and there will be centrioles on north pole and south pole. These centrioles, with the help of cytoplasmic fibers that are formed because of the cytoplasm, and these cytoplasm threads, all along the threads, the centrioles will pull these chromosomes up and down. So movement of chromosomes is possible because of centrioles, and each centriole is made up of 9 fibrils of tubulin protein. So tubulin is the protein, and the central part of centriole is called hub, and the peripheral fibrils are called spokes. So there will be 9 spokes. And then we go to the very important one, nucleus. Nucleus has highly extended, elaborate nucleoprotein fibers called chromatin, nuclear matrix, and nucleoli. The outer membrane is continuous with the endoplasmic reticulum and bears ribosomes. The chromatin materials change into chromosomes during active cell division. It consists of DNA and histone proteins. And as such, you may recollect, the nucleus was discovered by great scientist Robert Brown. It is a double membrane bound, dense protoplasmic body that controls cellular metabolism. It's the boss of the cell. Encloses all the genetic information because it has got chromosomes, and chromosomes have DNA, and DNA has got genes. So portion of DNA in fact is gene. So the genes contain genetic information, and it is able to transmit the characters or traits from one generation to another generation. The nucleus can be differentiated into following parts. One is the nuclear envelope consisting of 2 unit membranes, and the second one is nuclear matrix. That nuclear matrix is the network of fibrils function as scaffold for chromatin, and of course the nucleoplasm, the clear fluid containing substances for nucleotide synthesis, and of course all important nucleolus. Nucleolus again is responsible for formation of centrioles, and it plays a great role. Nucleolus plays a great role during cell division. And then of course all important chromatin, hereditary part of the nucleus, and is differentiated into heterochromatin and euchromatin. Basically, nuclear envelope maintains the shape of the nucleus and regulates the flow of materials. Nuclear pores are the passages through which the movement of RNA and protein molecules takes place. Nucleolus synthesizes and stores RNA. Heterochromatin is a genetically inactive and highly condensed region of chromosome with a tightly packed DNA. On the other hand, euchromatin is genetically active and rich in loosely packed DNA. And you may recollect, transcription occurs because of euchromatin. And then the chromosome part of it, when we talk of the chromosome part of it, as you see the disc-shaped kinetochores being present, you got nucleoplasm, nucleolus, nuclear pore, and double membranous nuclear membrane. and inside that you got all important chromosomes. On the basis of position of centromere, chromosomes are of the following type. Now I want you to focus on the position of the centromere. Basically, centromeres consist of this round structure, just like a rudraksha. You just take a rudraksha bead, and through the center of the hole you insert two threads, and that would be similar to the chromosomes. So centromeres consist of a complex combination of proteins and DNA. They are essential to the division of cells and ensure that accurate segregation of chromosomes take place. And studies have demonstrated that chromosomes without centromeres segregate randomly and are eventually lost from cells. In contrast, chromosomes that have multiple centromeres can be subjected to fragmentation. So based on the location of the centromere, chromosomes are of four types, as you are seeing. One is the metacentric chromosome. Meta means middle, center. Look at that position of the centromere. Metacentric chromosomes have the centromere in the center, such that both the sections are of equal length. You see this length of the chromatid and this length, and this whole chromosome as well as this whole chromosome length will be same. So human chromosomes 1 and 3 are metacentric, and then we come to submetacentric. Sub, as the name says, submetacentric chromosomes have the centromere slightly offset. You see here, it's slightly not in the center, slightly offset from the center, leading to a slight asymmetry in the length of the two sections, as you are seeing. Human chromosomes 4 to 12 are submetacentric. So in the case of 1 and 3, these are the chromosomes, and in this case 4 to 12 chromosomes in human beings are submetacentric. And the third one, acrocentric chromosomes. Acro means a tip. Acrocentric chromosomes have a centromere which is severely offset from the center, leading to one very long chromosome, as you are seeing, long section, and the other one is very short. And in human chromosomes 13, 15 and 21 as well as 22 are all acrocentric chromosomes. And finally we come to telocentric chromosomes. Telocentric chromosomes have the centromeres at the very end of the chromosome, as you are seeing here, almost a tip. And humans do not possess telocentric chromosomes, but they are found in other animals such as mice. So this is how the chromosomes play a crucial role, and they are important carriers of the genetic messages. And further, the chromosomes have non-staining secondary construction of at certain location. This gives a small fragment called satellite. And going ahead, we have got some cytoskeleton structures. You may recollect microtubules, microfilaments, and intermediate filaments are proteinaceous structures to support the extensive system of membranes. They collectively form cytoskeleton of the cell. Microtubules occur in cilia, flagella, centrioles, and basal bodies, mitotic apparatus, etc. Prokaryotic cells lack microtubules. So what is the microtubules which form the cytoskeleton? Microtubules. Tubules, T-U-B-U-L-E-S, microtubules. Okay, and the microtubules help in the spindle and astral ray formation during cell division. These form the cytoskeleton of cilia and flagella. So they help in cell division as well as in the formation of cilia and flagella. This is important again as far as your NEET exam is concerned. And then microfilaments are mainly composed of globular protein actin. So you got another cytoskeletal structure that is called microfilaments. microfilaments. And these microfilaments are mainly composed of protein, actin, but have filamentous protein, myosin also. So there are the microproteins contain two important proteins. One is the actin, another very important protein, myosin. And remember, I told you, if this is not present in required quantity, there is a genetic disease, myasthenia gravis, myosin. Okay, MYSOIN, myosin. Okay, intermediate filaments are non-contractile hollow filaments of acidic proteins. So microtubules help in spindle and astral ray formation. During cellular division, these form the cytoskeleton of cilia and flagella. On the other hand, microfilaments are involved in cytoplasmic streaming and amoeboid movements. That's paramecium and amoeba. And then the cilia and flagella are microscopic, contractile, filamentous processes of cytoplasm. The cilia and flagella have a fibrillar or microtubular composition. Their core is called the axoneme. The core is called axoneme, and usually it has 9 doublets. Okay, the core has got 9 doublets. That is, we can say 9 pairs, 9 doublets, and the core is called axoneme, and it has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. So you can say 2 plus 9 or 9 plus 2. Such an arrangement of axonemal microtubules is referred to as 9 plus 2 array. Okay, you can remember it as 9 plus 2 array. This is how the arrangement would have been there in the cilia and flagella. Both are capable of producing a current in fluid medium for locomotion and passage of substances, act as sensory organs, and perform many mechanical functions of the cell. And of course, the very important centrioles. These are 2 cylindrical rod-shaped microtubular structures near the nucleus. Centrioles lack limiting membrane and DNA or RNA and form spindle of microtubules. The mitotic apparatus during mitosis or meiosis. And of course, you must remember again this arrangement. In the case of cilia and flagella, it is 9 plus 2. In the case of centriole, it is 9 plus 0 array arrangement. Okay, this you must remember again, neat exam. The polymerized microtubules are for formation of spindle fibers and astral rays during mitosis and meiosis, and as I said, they help in pulling the chromosomes to North Pole and South Pole. That's how the 2N becomes N, and the N, this process plays a crucial role because the identity of the species depends upon the diploid or haploidicity of the organism. So thanks to centrioles, they determine the poles during cell division. Hence, the microprocesses, microtubules, cytoskeletal parts play a crucial role. Thus, I think we are winding up this chapter. Draw the diagrams, particularly plant cell, animal cell, label it,